Okay, what I think is the last video is about the human vaginal microbiota. This is something that hasn't been studied for quite as long as the colonic microbiota, and one reason that we should understand it better than we do is bacterial vaginosis, which is a more serious health problem than people give it credit for being. So I want to start by pointing out what we know about the microbiota and what they do and how our bodies try to control them, but then look at what goes wrong when we can't control them. So the vaginal microbiota are controlled in a big way by the host. And this is something you probably learned in A&P, that the epithelial cells secrete glycogen. Amylases break it down into sugars, and lactic acid bacteria, specifically lactobacillus, break down the sugars and secrete lactic acid. Lactic acid is toxic to a lot of bacteria. It is a strong enough acid that it keeps the pH something like 4, which is too low for most pathogens. So in addition to that, which protects us through making the mucus inhospitable to pathogens, also, the epithelium is secreting defensins. Those are antimicrobial peptides. And notice how that the total amount of defensins changes over the menstrual cycle repeatedly. Lactoferrin, I talked about a little bit when I talked about the innate immune system. You might go back and think about what lactoferrin does and why we care about binding iron. But day 7 of the menstrual cycle, it's high. Day 21, it's low, and then it comes back up. And the same pattern is followed by secretion of whatever antibodies the body is currently making, IgG and IgA. And lysozyme, the other enzyme I talked about that breaks down peptidoglycan, follows a pattern throughout the course of the menstrual cycle. And this is a simplification of these patterns. And I'll show you a more higher resolution thing on another slide. And yeah, this is that slide. So this is showing someone who studies defensins, those antimicrobial proteins, measured specific ones at different points of the menstrual cycle and showed that different defensins are released at different points of the menstrual cycle. And different other things, I don't know what those are, LFN and SLP1, I don't know what they are, but they are released at very specific times. So clearly, the body is trying to exert control over the microbiota in a big way that is tied to the menstrual cycle. And I want to show you this, it's gotta be 15 years old now, study, because it is fascinating. What these researchers did was ask 4 healthy women to basically give samples. They could figure out what bacteria were in their vaginal microbiota. And at that time, they were able to identify all of these. These are different bacteria, and they color-coded them. And the colors are going to be hard to tell. There are just too many colors in this diagram. But let me walk you through it. In this rectangle with all the funky colors, each column represents a sample of the vaginal microbiota from a certain day. And the different colors are the different percentage of the microbiota made up of those different bacteria. So it's always going to add up to 100. And let's just look at this woman's microbiota. I also should point out they keep track of when she was menstruating with these red bars, and they took samples for 15 weeks or 16 weeks. Well, this woman is mostly dominated by this red, which is one of the lactobacillus. And during menstruation, sometimes this green one becomes common. Always, the blue one is at a low level, the yellow one is at a low level, but the red one usually dominates between menstrual events. Now, all 4 of these were verified to not have bacterial vaginosis. So these are 4 healthy microbiota. Compare that first one to the one on the right. Well, this person is dominated by the red one all the time, except for one random time during the first menstrual cycle they followed. So this person has very low diversity of lactobacillus, where this person has a few, 1, 2, 3, 4, maybe 5 different main lactobacillus. And then down here, this person doesn't have as much of a clear correlation between a certain type and menstruation events. So this person has a diverse lactobacillus group, many different species, 1, 2, 3, 4, 5, 6, 7, 8, 9, something like that most of the time. Whereas this person had 1, this person had 5. And then this person is dominated by yet a different lactobacillus, and they seem to have, the only thing we could predict about them is that during a menstrual event, their main lactobacillus becomes less abundant and their lactobacilli become more diverse. The same thing happened with this person, but different species. And also notice how dramatic this is. After week 14, a different lactobacillus became the abundant one. So the point of this is that there are different patterns found in different healthy microbiota. There's not one we can point at, and if you wanted to say, I'd like to make a healthy microbiota, here's what it would have to look like, we can't really do that. We know there are different ways to be healthy, and we know there are different ways to be unhealthy. So that brings us to bacterial vaginosa, and this is a state where there's high diversity of microbes, and they are not lactobacillus. They tend to be obligate anaerobes. And at this point, the microbiota doesn't have the lactic acid bacteria. It can't convert the sugars and glycogen into lactic acid, so it can't keep the pH down around 4. It also can't prevent pathogens from surviving in the mucus. I mentioned chlamydia. Lactobacillus tie up essential nutrients and don't make them available for other microbes. But when we get rid of the lactobacillus, those nutrients are now available in the mucus for pathogens to use. That's nutritional immunity, like having lactoferrin, which ties up iron. And so in bacterial vaginosis, the clearest sign of disease used for the kind of the gold standard for diagnosis is a Gram stain of the vaginal smear where they can see bacteria adhered to epithelial cells. There should be separation between them. They should not be getting to where the cells actually are, but something is so wrong in bacterial vaginosis that the bacteria get to the epithelial cells. This is incredibly common, and we know the common symptoms and sign. They would be discomfort, a fishy odor, and discharge. But the main thing to know is that something like 80% of cases are asymptomatic. So in something like 80% of cases, you can find bacteria attached to epithelial cells, but the woman wouldn't even know there was a problem. But the complications of bacterial vaginosis still happens when people have the asymptomatic cases, and we'll get to that in a second. Treatments include antibiotics that try to kill those anaerobes, but that doesn't fix recurring bacterial vaginosis. Another more recent treatment is giving antibiotics to male sex partners, and that seems to give a slightly longer lasting treatment than antibiotics alone. So again, there are a lot of risk factors for bacterial vaginosis, but no one knows exactly why it happens. No one knows how smoking or multiple sex partners, or douching, or just having sex with someone who's not wearing a condom, with a man who's not wearing a condom. No one knows why that would lead to bacterial vaginosis. So there's no one major risk factor that predicts it, and no known cause, and there's no known cure. And the complications are the real story here. Pregnancy complications and preterm birth are risks of bacterial vaginosis, even when it's asymptomatic. And that increased susceptibility to STIs, including some viruses, and I do not know how this works. Chlamydia is not a virus, that's bacteria, but HIV, that's human immunodeficiency virus, HPV, human papillomavirus, both of those are more likely to happen when a person has bacterial vaginosis. So it is something I hope we learn about in more detail and eventually find an understanding of the cause and find a cure for it. Okay, it turns out that wasn't the last video. The next one will be the last video, and I will see you there.